Transduction is a type of genetic transfer of bacterial genetic markers from one cell to another mediated by a bacteriophage. Bacteriophages, or simply phages, are highly specialized bacterial viruses, meaning they only attack bacteria. These phages contain more parts of DNA or RNA that they can inject to the bacteria. Once injected, the virus dissolves and the injected genome takes its action in the cell. If the process results in bacterial death or bacteriolysis, we call it lytic phage, and the process involved is generalized transduction. Sometimes, however, instead of killing the bacteria, the injected genome gives the bacteria some special characteristics, like production of exotoxins, resistance to heat, and resistance to antibiotics. In this case, the phage is called lysogenic, and the process is referred to as specialized transduction. To further enhance our understanding, let's talk more about the lytic and lysogenic cycles and how bacteriophages reproduce. Let's put in mind that the transduction process always has to do with bacteriophage, which we described as viruses that attack bacteria. This process doesn't occur in animal viruses. So say for example, we have a bacterium. This is the bacterium right here, and this is the bacterial chromosome. This is the bacteriophage or simply phage, which is the virus that attacks the bacterium. This bacteriophage injects its DNA into the bacterium. This bacterium now has its own DNA and the DNA from the phage. Now, the host DNA is destroyed and degraded. The only remaining DNA now is the DNA injected by the bacteriophage. This event is followed by a synthesis in the host cell. Proteins are produced, basically the viral proteins. The phage DNA will code for production of new viruses. The newly produced viruses will exit the bacterial cell, causing the cell to lyse or break open, which results to the destruction of the bacteria. This is called the lytic cycle. On the other hand, let us talk about the lysogenic cycle. The preliminary steps are the same with the lytic cycle, where a virus or phage will attack the bacterium. The virus will inject its genome here and incorporate its DNA to the host bacterial cell. But the difference here is that in the lysogenic cycle, instead of the DNA from the phage destroying the host DNA, it actually integrates itself into the chromosome of the host DNA. So now, the difference is that there is no destruction yet. The advantage here is that these cells are now capable of reproduction at much slower rate than that of the lytic cycle. Every time the cell divides, you can see that a fragment of DNA from the phage, which is called the prophage DNA, is going to be carried from generation to generation. All the newly produced bacteria will now carry this piece of viral DNA. At some point, when this host cell becomes stressed or exposed to unfavorable conditions, a stage called phage induction happens. In this stage, the DNA fragment from the phage pops out and now the cell grows through the lytic cycle. Now, the second half of the lysogenic cycle is basically the lytic cycle, where again, it's gonna destroy its host DNA, produces viral proteins, and exits and destructs the cell.